Okay, hi. Well, it's always a great pleasure to introduce a friend and colleague. I had the privilege of collaborating with Sue on her work on the relationship between the gut and airway microbiome and lung diseases, especially asthma and COPD. But her work extends far beyond that interaction. So I'm delighted that she's agreed to give this presentation today. Sue is a graduate of the University of College Dublin. She did her postdoctoral studies at Stanford, and then we were lucky she came to UCSF to pursue her career thereafter. She's now the director of the Benioff Center for Microbiome Medicine, and she and Jennifer Doudna, Nobel laureate from UC Berkeley, have together won a major TED Audacious Award to investigate whether applying CRISPR technology to the genes of the bacteria that Sue has identified in the gastrointestinal tract of children predisposed to develop allergic asthma. If those genes can be modified by CRISPR, the bacteria are reintroduced and lower the risk of those children. This is a disease that affects 300 million people, at least, on the planet, and has been increasing in prevalence for 60 years or 70 years now. So it's an important childhood disease, and one affecting adults, of course, as well. So I think I should leave it there, give time to Sue to make the presentation. So Sue, thank you.
Great, thank you so much, Homer. And I believe, Luz, you may need to start my video because it's not allowing me to do it on this end. There we go, perfect. Right, let me get my slides up and get started. Thank you so much, Homer, for the introduction and more broadly for the invitation to be here and join you today. My group works on the role of the microbiome, the communities of bacteria, fungi, and viruses that call the human body home, and how those microbes may drive chronic inflammatory diseases. So as Homer mentioned, I work in a number of different disease areas, including inflammatory bowel disease, and more recently we've branched into periodontal disease, which is an inflammatory disease in the oral cavity. But I have to say my kind of first love has always been allergy and asthma. And I think that's in part because I got to do a lot of this work with Homer as a collaborator for many, many years. And it's been just an area that I've been intrigued by. And I do think that we're starting to find some of the answers as to the origins and the genesis of the disease. And that's the work that I will share with you today. And just by way of disclosure before I get started, I co-founded a company called Shield Therapeutics. I'll briefly mention what they're up to at the end of the presentation. I also consult for the Atria Institute in New York and for Sanofi, but I won't be talking about any of that work here today. But to get started, I am by trade a microbiologist, and I was trained in the very traditional ways of microbiology, like you see on the agar plate side of the screen. So back in the day when I trained, we would pluck individual microbes out of human biospecimens. We grow them in isolation on agar plates in the lab and study them in isolation. But nothing could be further from how microbes actually exist in nature, and not just in humans, but in all kinds of terrestrial and aquatic ecosystems. Microbes exist in diverse communities that interact with each other. Microbes are capable of communicating with each other by secreting soluble small molecules that allow other microbes to understand which microbes are in their environment and how they're behaving. And so many years ago, luminaries in the field of environmental microbial ecology recognized that the number and types of different microbes that you could observe under a microscope, for example, in a couple of milliliters of seawater, could never be recapitulated on the agar plate under conventional culture conditions in the lab. And so they set out developing these culture-independent approaches to identify microbes in diverse communities without ever needing to culture them. And the workhorse in the field is really these sequence-based approaches to identifying which microbes are present and what they're doing, what they're expressing, the genes they're expressing. Much of this work is facilitated through profiling of DNA extracts from samples. So a sample has its DNA extracted from it, and remember that DNA can come from any of the microbes that are present in the sample. Then there's a number of different approaches that can be taken. The bacterial exclusive gene, 16S ribosomal RNA, the ribosomal RNA gene can be amplified, or there's another region of the genome in fungi called the interspacer region 2. Amplification of those genes or those regions allows you to take that piece of DNA from any microbe that's in a mixed species community. And then by virtue of sequencing that pool of amplicons, you can get a sense of which microbes are present, kind of like a fingerprint of the bacteria or fungi that are present in a sample. But that just tells you who's there and their relative concentrations. If you want to delve deeper and understand the genes that these microbes encode, you can take that extracted DNA and just sequence the whole pool of DNA. That allows you to understand the genes encoded by the diverse microbes that are found in that sample. And that allows us to assess functional capacity or functional genes present in microbiomes. If you preserve samples appropriately, you can extract RNA. And this allows us to assess what genes are being transcribed by multiple different microbial species within a microbiome or a mixed species community. And then mass spectrometry approaches have really amplified our capacity to understand how these microbiome communities are behaving, how they're functioning, and how they're interacting with the host. And that's obviously particularly interesting in the case of the human microbiome. If we profile proteins with mass spectrometry, we can get a sense of which ones are related to, for example, clinical outcomes or exposures. And the same is true with metabolites. These are basically biochemicals produced by microbes and the host in biospecimens. But leveraging this kind of data can really get us down to the kind of molecular lexicon of microbial host interactions that allows us to identify small molecules that are mediating these interactions between microbiomes and their host. So this is an incredible suite of tools that's also enhanced by a number of model systems that we use in the field to really allow us to go from soup to nuts and examine human populations and break down mechanisms by which microbes promote immune dysfunction to drive disease development in human populations. And the application of these tools over the last 15 years or so, because this is still a new field, has allowed us to understand that as humans, we're really superorganisms. We are made up of microbial and human cells. And in the gut microbiome, especially in the distal gut, we house trillions of microbial cells. And think of it this way, there are hundreds, and in some cases thousands of species within the gut microbiome, each species having its own genome and contributing a suite of genes to the overall function of the gut microbiome. The genes in the gut microbiome dwarf that of the human host by about 150 to 1. There's millions of microbial genes in the gut that govern how we metabolize food and how these microbes interact with the host immunity. Much of host immunity is driven by interactions in the gastrointestinal tract. The large surface area colonized profoundly by microbes. But I think what's most exciting about the human microbiome field is that we can manipulate the human microbiome. It really is medically accessible, and I think more manipulable than the human genome, though some may argue with the advent of CRISPR that is not necessarily the case now. And we've been really interested in applying these tools to understand asthma, especially childhood asthma, because it really is a disease of westernized or industrialized nations. And you can see that from this really nice map of the prevalence of asthma across the globe. And you can see that it is really concentrated in those countries where we've become highly industrialized. And it really is reaching really epic proportions in pockets of Western Europe where almost 20% of the population are diagnosed with asthma. And while we have some work going on in established disease, I'm really going to focus on a very major interest of ours in the group, understanding the developmental origins of allergy and asthma. And the first thing I should state is that the human microbiome is not static, and it develops over the first 3 years of life very rapidly. But it continues to evolve throughout life based on environmental exposures of an individual. And so knowing that, that suggested to us that really understanding the microbiome may prove to be the ticket to understanding asthma and allergies in large tracts of the human population. The other key thing to note is that over the first 3 years of life, the gut microbiome develops quite rapidly. This is a critical period in which we accumulate microbes and their genomes, and therefore their functional genes, into the gut microbiome. And it just so happens that that parallels a critical window in immune development. We know from mouse studies that immune training and priming that occurs in early postnatal life cannot be undone in later adult stages of life. So really suggesting that these initial encounters with microbes in the earliest stages of postnatal development may be really critical to shaping the trajectory and risk of disease development in childhood. The other thing to point out was that we really got to stand on the shoulders of giants as we engaged in this field. There are many epidemiological studies that have demonstrated and validated that early life exposures, either in early infancy or in the prenatal period, including things like cesarean section, formula feeding, and antimicrobials, increase risk of allergy and asthma development. Whereas exposure to more traditional exposures like livestock and even pets, dogs and cats in the home, decrease risk of allergy and asthma in childhood. We know that these are factors and exposures that shape the types of microbes that exist in the gut microbiome. And so this suggested to us that in order to understand a disease that is typically diagnosed in childhood, we may have to dial back the timeline and begin looking in early infancy, in the early postnatal period, when the microbiome is developing, to understand whether there are microbial predictors and microbial drivers of allergy and asthma risk in later childhood. And so the first study that allowed us to examine this was actually a trial led by Michael Cabana when he was at UCSF and led by Julia Derrick and Nicole Kimes, who were both postdocs in my lab at the time. And Michael was studying whether supplementation with a commercially available probiotic, Lactobacillus rhamnosus GG, daily for the first 6 months of life in high risk for allergy and asthma babies could prevent atopy and wheeze development in later childhood. So he was collecting stool samples from these babies at birth, 1, 3, 6, 9, and 12 months of age, and from a healthy control cohort at the same time points. We took these samples and we applied 16S ribosomal RNA sequencing to examine the bacterial communities that were present. And we used metabolomics or mass spectrometry to examine the small molecules or metabolites being produced by these microbiomes. And the first thing that was really striking to us was that the meconium microbiota of high-risk babies was significantly different from that of their low-risk counterparts. This is a principal components plot, and this is how we visualize these kind of very high-resolution bacterial barcodes or bacterial fingerprints that we produce from these sequencing approaches. Each spot represents a profile of bacteria found in an individual infant in this study, and the distance from one spot to another spot tells you how similar those microbial or bacterial profiles are. Here you can see that the healthy control population in green is spatially separated on the diagonal from the high-risk placebo group here in red, indicating that there is significant difference in the microbiota of these infants at birth before they even start to develop their microbiome. And that difference in microbial composition at birth is maintained throughout the first year of life. Here we can see the high-risk group of infants in the placebo arm of the trial, and these are high risk by virtue of having at least one parent who had allergies or asthma. But you can see that it really diverges in terms of the pace of diversification of their gut microbiome over the first year of life compared to their healthy counterparts. So this suggests to us that there are strong selective pressures in the gut of these high-risk babies that prevent accumulation of beneficial microbes that prevent allergic asthma development in later childhood. And we had additional data that really, from a completely independent cohort, that really allowed us some insights into how this is happening, what is happening with these gut microbiomes, and how they may be modulating immune function. This was the Wheels cohort led by Dennis Ownby and Chris Johnson at Henry Ford Hospital. And the work I'll present was led by Kei Fujimura, who was then a postdoc in my lab. The Wheels cohort is a racially and socioeconomically diverse population, birth cohort population in the Detroit greater metropolitan area. And we took infant feces, stool samples from 1-month-old infants. And here again, we performed 16S profiling to examine the bacterial community composition and ITS2 profiling to examine the fungal communities that were present in 130 infants in this study. And because this is a birth cohort, Dennis and Chris had followed these children out into childhood and knew whether they developed A to B or allergic sensitization at 2 years of age or asthma at 4 years of age. And the first thing we did with our 16S bacterial profile data was took kind of a hands-off approach. We asked an algorithm, a Dirichlet multinomial mixture model, whether there were distinct bacterial community compositions in these 1-month-old babies. And the answer was yes. And the second answer was there were 3. There are 3 different distinct bacterial community compositions in these 1-month-old infants that we called neonatal gut microbiome 1, 2, and 3. The next question is whether having a different gut microbiome at 1 month of age altered your risk of developing A to B or asthma years later in childhood. Indeed, were there early microbiome signatures that increased risk of disease development? And again, the answer was a resounding yes. Babies who at 1 month of age had the NGM3 gut microbiota composition were at significantly higher risk of developing A to B at 2 years of age and asthma at 4 years of age. And this was actually the first time that it had been shown that there was a very early life gut microbial signature that was associated with increased risk of childhood disease development years later when disease is diagnosed. So what was different about this NGM3, this distinct gut microbiota in these infants? Well, it was depleted of a whole range of bacteria. Again, consistent with this idea that there's a loss of diversity that is really kind of characteristic of these infants in early life that are at high risk. We saw an enrichment or an overgrowth of particular fungi, specifically Candida and Rhodotorula, and both of those have been shown in mouse models to promote allergic inflammation when they're introduced into the gut microbiome. So pro-allergic fungal organisms were enriched in these 1-month-old infants who are at higher risk of developing disease. Because there was really a massive depletion of bacteria, we predicted using a software that these infant gut microbiomes in these higher risk babies were really functionally deficient. And the majority of genes and pathways that were missing from these 1-month-old babies were in pathways that allow microbes to metabolize substrates. So this gave us a clue that perhaps what these infants have at 1 month of age is a really distinct metabolic machine a microbiome that is pumping out a different suite of biochemicals to engage with immune function and train immune function in very early life. And indeed, in applying mass spectrometry to these samples, that's exactly what we found. The metabolic milieu in these high-risk babies was significantly different from that of the lower-risk infants. And specifically, some of the features that were characteristic of these high-risk babies' metabolomes was that they were highly enriched for simple sugars, which we're now beginning to understand are inflammatory. That's also true for monohydroxy fatty acids, which we found to be enriched in these infants as well. And they were depleted of a range of anti-inflammatory lipids, including polyunsaturated fatty acids, or PUFAs, and steroid metabolites that we know downregulate or reduce inflammation. So this suggested to us that we actually have maybe a kind of an inflammatory microbiome at 1 month of age in these babies who go on to develop allergies and asthma later in childhood. But we only had stool from these babies, and the only way we could really test this was coming up with a kind of an outside-the-box assay. We took the feces of these 1-month-old infants, and we took out all of the cells from that feces, all of the microbes, any mammalian cells that were present. So in essence, all that we're leaving are those metabolites that are being produced by the microbiome of these babies. And we took that cell-free fecal extract and we co-incubated it with dendritic cells that were purified from an adult healthy donor. And dendritic cells are a key group of immune cells that present antigens to naive T cells and really informs them what they become when they grow up and mature as T cells. And T cells can mature into a number of different effector phenotypes: Th1 cells, which can be inflammatory, Th2 cells, which are inflammatory and are the key group of T cells that mediate allergic inflammation. Th17 cells are T regulatory cells. And T regulatory cells are the critical group of immune cells that put the brakes on allergic inflammation. So we were particularly interested in these Th2 and these T regulatory cells, primarily because in children with asthma that's been diagnosed, one of the key characteristics, or the key immune characteristics that they have, is they have reduced numbers of T regulatory cells producing a cytokine or a small molecule called IL-10 that reduces inflammation. And instead, they have a large expansion of these Th2 cells that produce these inflammatory cytokines or molecules called IL-4, IL-5, and IL-13. And these children also have elevated concentrations of an immunoglobulin in their circulation called IgE. And this is kind of the hallmark of allergic asthma immune dysfunction. So with our assay, we asked, what can the products of these infant gut microbiomes promote in terms of T cell effector phenotypes? And what we found was that the NGM3, or high-risk gut microbiome, could essentially recapitulate the critical features of immune dysfunction associated with allergy and asthma in a test tube. We saw an expansion of the Th2 cells, an increase in the inflammatory cytokine IL-4, and a decrease in the anti-inflammatory T regulatory cells. So this told us that we have a pathogenic inflammatory microbiome at 1 month of age in babies who have a different microbiome and who go on to develop allergies and asthma in later childhood, suggesting that really the genesis of the disease is happening in the earliest stages of postnatal life, at least. But we wanted to dig down deeper and find out, what was actually driving this immune dysfunction. So we took all of the metabolites we had and really broke them down into groups of co-associated metabolites. So when we see these metabolites, we always see they co-associate with other metabolites. We then tested whether any one of these metabolic modules was found to be increased in the NGM3 group, and we found only one of them. This one group of co-associated metabolites that I'm showing here was found to be increased in the high-risk NGM3 group. And what I'm showing here is a plot that shows module membership. That's a measure of how interconnected each metabolite is in this group of co-associated metabolites and significant. So we could see that the top hit here was a lipid called 12-13-dihome. It's most interconnected to all of the other metabolites in the module, and it's significantly associated with the NGM3 group. And when we quantified this lipid, we saw that indeed it's significantly enriched in that high-risk group of infants. So we next went on and we repeated the same assay with the immune cells again, and now asked whether this lipid could recapitulate any of the features that we'd seen initiated with the fecal extracts. And indeed, what we found was that this lipid, 12-13-dihome, in a dose-dependent manner, can reduce those anti-inflammatory T regulatory cells and decrease their capacity to produce IL-10. So we now found a component in this metabolic milieu that appears to be driving a critical component of immune dysfunction that underlies allergy. So Sophia Levin in my group wanted to see whether this worked beyond a test tube. And so she took mice and she broke them into different groups. And these are mice in which we can induce airway allergic inflammation by challenging their airways with cockroach antigen, or CRA challenge. And she peritoneal delivered this lipid, 12-13-dihome, to one of these groups of mice to ask specifically whether having 12-13-dihome in the system exacerbates airway allergic inflammation. And the resounding answer was yes. These are control animals that we've not manipulated. These are animals that we've induced airway allergic inflammation. And now you can see in the air spaces in the lung, there's a lot more of these nucleated cells. These are inflammatory cells that are circulating around the air spaces. And this is true also around the blood vessels. This is the group of mice that received the lipid 12-13-dihome. And as you can see clearly, that now we have massive inflammatory infiltrates around the air spaces and the blood vessels. And now the air spaces are, they're occluded with pink staining mucin. So these are really inflamed airways in these animals, just by virtue of the fact that we've provided them with this one lipid. She also validated these observations at a molecular level. She showed that only the mice that received the 12-13-dihome and had cockroach antigen challenge, they were unable to mount this T regulatory response in their airways. So again, this lipid mutes the capacity to dial back down inflammation, allergic inflammation, once it's initiated in the airways. And she found that in the serum, or the circulation of these animals, they had more IgE, that immunoglobulin that's associated with allergy. So really demonstration that this lipid is a pro-inflammatory allergic lipid that really enhances allergic inflammation in the airways. She next addressed the hypothesis that this lipid is actually being produced by the microbes in that perturbed 1-month-old gut microbiome. And to do this, she took feces from 26 babies in the Wheels cohort. She performed shotgun metagenomics. Now we're delving deeper to look at the genes encoded by these microbes. And she performed an approach called shortbread that allows you essentially to go fishing for genes in that mix of genes that are identified by sequencing analysis. And she was specifically looking for epoxide hydrolase genes. This is the gene that catalyzes that terminal step in converting an epome into the 12-13-dihome. And what she found was that there were significantly more epoxide hydrolase genes in 1-month-old infants who went on to develop atopy or asthma compared to their healthy counterparts. She also showed that having more of these genes in the gut microbiome of these babies associated with having more 12-13-dihome lipid in their feces. So this was a good start. But having a gene that looks like an epoxide hydrolase doesn't tell you that it actually makes 12-13-dihome. And so she dug deeper into this data, and she found the 11 most prevalent epoxide hydrolases in these infant gut microbiomes. And you can see from this heat map, they're predominantly found in the atopic infants in the study. She cloned each of those genes into E. coli and then performed a simple functional assay. Are all of these genes epoxide hydrolases? Can they convert glycidol to glycerol? And then B, which ones can actually produce 1213 dihome from its substrate 1213 epihome? She also asked whether any of these could produce 910 dihome as well, which is another epoxide hydrolase functional attribute. And she found that all of them were epoxide hydrolases. So the metagenomic analysis had worked very well. 2 of them could produce 910 dihome, and 3 of them, encoded by Enterococcus faecalis and Bifidobacterium bifidum, could produce the specific lipid we were after, 1213 dihome. So Sophie next performed really the take-home experiment for this study. She took the E. coli that could now specifically produce 1213 dihome, and she fed them orally to groups of mice. So here we have control mice. We have mice that get the epoxide hydrolase producing and 1213 dihome producing E. coli. And in 2 of the groups, she also additionally challenged their airways with cockroach antigen. So the test here is if you have the microbial gut capacity to produce 1213 dihome, do you see increased concentrations of 1213 dihome in the circulation of those animals? And do you see a reduction of regulatory T cells in their airways? And the answer was yes. So if animals receive the capacity to produce 1213 dihome by this E. coli engineered to produce it, we saw significant increases in that lipid in the serum, and actually, though I'm not showing it, also in the airways of those animals. And as we hypothesized, we showed that when we challenged these animals with cockroach antigen into their airways, the animals that can produce and actually do produce high concentrations of 1213 dihome fail to mount an anti-inflammatory T regulatory response in their airways. So this demonstrated to us for the first time that gut microbial production of a lipid metabolite, 1213 dihome, can alter immune function in the airways at a remote site and exacerbate allergic inflammation in an animal model. The other key thing from this study was that when we looked in 2 independent cohorts, we showed that every doubling of the epoxide hydrolase gene copy number in these infants in their feces correlated with an increase in risk of asthma or allergy development. And if we kind of look at the copy numbers, infants that just have 4,100 copies per gram of feces of these bacterial epoxide hydrolase genes that can make 1213 dihome have an odds ratio of 15 of developing allergies and asthma. It gets even worse if you have high concentrations of the actual product of those genes, the 1213 dihome lipid. So for every nanogram increase in this lipid, in feces of these babies, you significantly increase the odds ratio of developing asthma and allergy. And infants that have 2,000 nanograms of this lipid in their feces have an odds ratio of 55 of developing allergy and asthma. And some of these children had double this again, had 4,000 nanograms of this lipid per gram in their feces. So this study told us that there are fecal microbially derived metabolites in very early life, in infancy, that promote central aspects of immune dysfunction associated with allergic inflammation and asthma development. But we were particularly interested in how this is happening and where these microbes are coming from. Remember, I showed you that in meconium samples, we already see that these babies have a different gut microbiome and follow a different trajectory of microbiome development over the first year of life. And we know from multiple studies in the literature, quite nicely summarized in this graphic, that the majority of microbes that are found in the infant, on the infant skin, in the gut, in the oral cavity, everywhere in the infant, come from their mother through vertical and horizontal transmission. And so that suggested to us that to really understand the microbial dysfunction that's occurring in very early infancy, we perhaps have to dial the timeline back into even earlier human developmental time and examine the prenatal period. And I should point out that many of the factors that relate to risk of allergy and asthma development in childhood also are related in the prenatal period. So again, exposure to livestock and cats and dogs in the prenatal period protects against allergy and asthma development, while things like antimicrobial administration increased risk. So suggesting to us that perhaps the maternal microbiome in the prenatal period is key to setting up the microbiome in very early infancy and the trajectory of childhood disease development. And we're particularly interested in understanding this because we want to know when to intervene to try and change this trajectory to prevent disease development. And so one of the studies we were engaged with, this was led by Katie McCauley when she was in my group, and this study was a harmonization of data generated from 2 independent cohorts within the ECHO Crew consortium. And these were vaginal samples collected at 35 to 37 weeks gestation, so in the late prenatal period, and paired infant stool samples collected at 1 to 2 months of age from 172 babies. We had maternal exposures and factors measured, and we had the children's factors, including an early marker of allergic disease development, the total serum immunoglobulin E, or IgE, at 12 months of age or 1 year of age in these babies. So babies that have high concentrations of IgE at that time in early life are really on the trajectory for atopic disease and asthma development. In addition to examining all of these samples with 16S bacterial profiling, we also dug deeper and examined them with shock and metagenomics in a small subset of mother-infant dyads to get at microbial genes and pathways that may be indicated as contributing to early markers of allergic disease development. And again, we take a kind of an agnostic approach to our data analysis. We don't go in with biases as to what we expect to find. And again, we asked an algorithm, are there distinct prenatal vaginal microbiota structures or compositions that we can observe in this cohort? And the answer was yes. There were 4 distinct bacterial vaginal microbiota states or clusters that we found. That's pretty well known across vaginal microbiota. There's actually 6 that are known, but in the prenatal period, it appears that really 4 of these are predominant, at least in the population that we studied. Interestingly, these vaginal microbiotas related to the health status and the environmental exposures of the pregnant individual. For example, those with vaginal cluster 1 or VC1 had significantly increased exposure to pigs in a farming environment, were more likely to be depressed, and had infants who had fewer siblings. You can see that the VC4 community composition here, these were pregnant individuals who were much less likely to have worked on a farm or have farming exposures, including farm feed, and they had increased maternal stress. So this suggests to us that health status of the mother and her environmental exposures may shape her microbiome during the prenatal period. We next asked whether the vaginal microbes that are present in the prenatal period are shared with the infant gut microbiome, essentially examining transmission of microbes between mother-infant pairs. And this plot that I'm showing is really a summary of 172 mother-infant dyads. Each column is a mother-infant dyad, and each red rectangle is a bacterium that's found both in the mother's vaginal microbiota and in her infant stool sample. And so you can see that there's some organisms that are more prevalently transmitted from mother to child, for example, Staphylococcus, Bifidobacterium, and Bacteroides. But our question here was, is the variance in microbiota transmission from mother to child related to aspects that we care about in the study? And the first question we had was, does the types of organisms that are transmitted relate to the type of vaginal microbiota the mother has? We're particularly interested in that because environmental factors seem to shape those microbiomes, or at least relate to the types of microbes that are present. And that was a resounding yes. So it matters which vaginal microbiota you have. That dictates or is associated with the types of microbes that are transmitted to the infant gut microbiome. Key to this was the mother's eczema and asthma status in the prenatal period also related to the types of microbes that she transmits to her infant. Her pre-pregnancy BMI also relates to these transmittable organisms, suggesting that perhaps obesity phenotypes may be related to transmission of microbes from mother to child. And critically, the infant total IgE, so that early marker of allergic inflammation, related to the types of microbes that are shared between mother and child across this cohort. Infant birth weight also related to the transmittable organisms, as did mode of delivery, raising the intriguing possibility that microbial transmission patterns may influence early life events like birth weight and mode of delivery. But we wanted to delve a little deeper, and though this is a very small number of samples that we examined, we asked which were the microbial genes and pathways that related to detectable IgE at 12 months of age in this cohort. So this is a subset of 12 mother-infant dyads where we've examined both the maternal vaginal microbiota and the infant stool samples with shotgun metagenomic analysis. And now we're looking at all the genes and pathways. And we looked specifically at the genes and pathways that were shared between mother-infant dyads. And we used artificial intelligence or a machine learning algorithm to tell us which of these genes and pathways related to the presence of detectable IgE in these babies. And this threw up a really interesting subset of microbial functions. And I want to focus first on those that were associated with no detectable IgE. So these are babies who do not go on to have early markers of allergic inflammation. These babies inherit microbial capacity to degrade histamine. I'm sure all of you know that if you've got allergies, one of the first approaches to deal with that is to take an antihistamine. These babies inherit microbial capacity to regulate and degrade histamine in the system. They also inherit microbial capacity to metabolize tryptophan, a potently immunomodulatory molecule that has been implicated in allergic disease development. Critically, we see that babies who don't develop early markers of allergy inherit microbial capacity to metabolize stress hormones, noradrenaline and adrenaline, suggesting that their microbiome in the gut can regulate stress hormone concentrations. An intriguing observation, given that we've seen that one of the microbiota compositions in the vaginal tract that transmits microbes is associated with stress in the mother. And then lastly, these babies also inherit microbial capacity to produce fatty acids, several of which we know can be anti-inflammatory. On the flip side, the babies who did develop early markers and detectable IgE by 12 months of age, indicating that they are allergic, inherit microbial capacity to produce for glucose-6-phosphate isomerase. This converts glucose-6-phosphate to fructose. Bear that in mind, and I'll tell you more about that in the next slide. They also inherit microbial capacity to produce glutamate. And while glutamate is more regularly known as a neurotransmitter, it's also more recently been found to activate T cells. And then lastly, we saw indications that this is a pathogenic microbiome. It's enriched for things like prepillin peptidase. Pili are microbial appendages that drive inflammation from host responses. And these observations were really critical when we saw some more recent data that has come out showing that fructose, which can be made by that glucose-6-phosphate isomerase, reprograms oxidative metabolism in a glutamine-dependent manner, glutamine comes from glutamate, to support lipopolysaccharide-induced inflammation. So what this suggests to us is that we have a pathogenic inflammatory microbiome in these babies that is producing metabolites like fructose and glutamine to enhance the inflammatory response to microbial presence. And obviously the microbes that are there are immune to that inflammatory response. But this is what we think is preventing the accumulation of commensal organisms into these gut microbiomes over the first year of life in these infants. And really this data provides what to me is the first evidence for an inherited microbial genetic risk for allergy and asthma heritability. We know that there's specific genes that are human genes that increase the risk of wheeze, but they really don't explain the full degree of risk in the human population. And I think that's because we're missing many of these risk loci because they are actually microbial risk genes that contribute to disease development. And I'm just going to spend the next 5 or 7 minutes going through one other study that we've published relatively recently in thinking about how non-asthmatic mothers may promote asthma development in their offspring through epigenetic modifications that influence microbial development in early life. And so this study was based off the observation by our collaborators that the ratio of interferon gamma to IL-13 in the circulation of non-asthmatic pregnant individuals relates to asthma development in their offspring. And remember, immune response is in large part governed by the microbiome in an individual. And so here you can see that those who have higher concentrations of IL-13 and lower concentrations of interferon gamma have the highest risk of childhood asthma development in their offspring aged 2 to 9 years of age. So we wanted to fill in the gaps in this study. And to do this, we took 155 mother-infant dyads from the original immune infant study, or the IIS study, in which the original observations were made. We knew the interferon gamma IL-13 status of the mothers. We generated an epigenetic profile for the cord blood mononuclear cells of those individuals. We examined the nasal microbiota over the first 3 years of life in these infants, and we knew clinical outcomes of asthma at age 2 to 9 years of age. And this is work with the long-term collaborator Donata Vercelli at University of Arizona, and Avery DeVries, Katie McCauley, and Deb Stern contributed to the study as well. The first question we had is, in this subset of 155 mother-infant dyads, do we see the same relationships that we saw in the much broader population? And the answer was yes. We saw that the maternal interferon gamma IL-13 ratio relates to the risk of childhood asthma across the 182 individuals we examined, but also specifically it becomes more stronger in the 155 that we studied for this investigation. Here you can see again that the ratio relates to childhood asthma risk. And so we saw that in those that are in the other 3 quartiles of interferon gamma IL-13 ratio, that they really were all significantly lower than those in that top quartile with the highest IL-13 and lowest interferon responses. So we grouped all of these 3 together as a comparator group to the first quartile, or Q1. We asked what epigenetic marks in cord blood mononuclear cells related to a low versus high maternal immune status or interferon gamma IL-13 ratio, and found that there were a large number of methylation marks that significantly differed based on the mother's immune status in the prenatal period. We again took a data reduction approach, as we've done in other studies, and we asked how these methylation marks kind of co-associated. So we broke them into groups of methylation marks or epigenetic modifications that consistently co-associate. And we found that one group of 578 methylation marks, significantly related to asthma outcomes in the offspring of these pregnant individuals. I don't expect you to be able to read all of this, but these basically are the methylation marks in cord blood mononuclear cells that in that module that relate to asthma development at age 2 to 9 in the infants, in the children in this study. What I do want you to note is that in purple, I've called out every one of these genes or loci that are associated with response to microbes. And so what you can clearly see is that there's a significant enrichment in methylation of genes associated with antimicrobial responses. And that suggested to us that perhaps in these infants who are at high risk of asthma development, these epigenetic modifications perhaps mute their capacity to respond to microbes, which would allow pathogenic microbiomes to develop in the gut and in the airways of these children and asthma development later in childhood. And we found that that was exactly the case. Here I'm taking what we call the turquoise module eigenvector. I'm basically taking a vector that represents all of these methylation marks, and I'm correlating it with the capacity of cord blood mononuclear cells to mount an IL-6 or an inflammatory response to lipopolysaccharide. And I find that there's a significant and strong correlation with the methylation marks in this module and the capacity to mount IL-6 responses. So this suggested to us that in those children, as you can see in red, who go on to develop asthma, you can clearly see that they are unable to mount an inflammatory response to microbial products when they have these methylation marks. So to test our final part of the hypothesis that this methylation profile related to a pathogenic microbiome development, we examined longitudinally collected airway samples from these children. These were the only samples that we have. And we examined their microbiome development over time. And as you can see, those in the high risk first quartile show a distinct trajectory of airway microbiome development compared to those in the low risk quartiles shown in black. And what it demonstrated to us is that those who are at highest risk of allergy and asthma development based on the maternal immune status start life with Haemophilus in their airways, and by 3 years of age tend to have Moraxella in their airways. And these, we and many other groups have now shown, are predictive microbial colonization patterns that are predictive of asthma development. So it really suggests to us, as we take all of this data together, is that maternal immune status, which we assume is largely driven by her microbiome in the prenatal period, and specifically the interferon gamma IL-13 ratio of pregnant individuals, influences the methylome of cord blood mononuclear cells, so essentially the fetal immune response, and mutes the capacity in those who are at high risk, mutes the capacity to mount an inflammatory response against microbial products. This is associated with development of a pathological microbiome in the airways of these children and asthma diagnosis in later childhood. And we believe that the same holds true in the gastrointestinal microbiome, but in this case, we only had airway longitudinal samples to study. And so if we take everything that I've shown you together, what it suggests to us is that prenatal exposures impact the microbiome of the mother during the prenatal period. We think it also impacts her immune status, though we've yet to show that definitively. And that the immune status and the microbiome of the pregnant individual in the prenatal period dictates the microbes that are transmitted, the epigenetic marks that are present at birth, and we also believe immune functions that are transmitted through breast milk, for example. And this in turn governs microbiome development in the earliest stages of postnatal life, which trains immunity in that critical first few years of life and ultimately governs childhood health outcomes in these individuals. And so what are we doing about it? Well, one of the things we can do is intervene. And one of the ways we can intervene is by using multi-species microbial consortia to reintroduce or to introduce the microbial functions that we know are missing throughout the entire first year of life in children who are at high risk of asthma and allergy development. And we want these to engineer microbial metabolic productivity in the gut, to train immunity, restore barrier integrity, and promote competitive exclusion of pathogens and allow for commensals to accumulate into the gut microbiome over the first year of life. And this, we believe or hope, will prevent allergic disease development. And this is actually a trial that just finished enrollment very recently, the ADORED trial, led by Shilta Therapeutics that I mentioned at the outset of the talk. And so all that remains for me to do is thank an incredible group of people that I get to work with. This is clearly interdisciplinary research. I am incredibly indebted to my mentors, my collaborators across the country, and the National Institutes of Health for their long-standing support of our program. And with that, I thank you for your attention.